Good morning, grade 11 learners of South Africa. What are we going to be doing today? Well, we're looking at plants, and we're looking specifically at structure of plants. Plants have an internal skeleton, just like us, providing strength and support. Now, what makes up that internal skeleton is going to be the xylem tissue, very important, but it also transports water. So it has double function or a dual function. Then we have the sclerenchyma tissue. Now, sclerenchyma tissue is tissue that is very, very strong. It's filled with lignin and suberin, and it makes it nice and firm. It's got big, big, big cell walls. Okay? And then we have water and the turgidity of the cells. So the water that's in the cells keeps the cells all nice and firm. And you will have noticed that if you have flowers, you pick a flower and it's got leaves on it, eventually as the flower dies, the leaves also just wilt and die. Now, when they start to wilt, they've actually lost too much water. They're losing more water than they can absorb, and that causes them to then wilt. So it's the water in the cells that gives them that structure. All right. As we move down, we say, right, we've got plants have an internal transport system. So their internal structure is the xylem and the sclerenchyma and the turgidity of the cells. And their internal transport system is the xylem, which transports water and mineral salts up the plant. You've got the phloem, which transports mineral salts and food up, and water, and down the plant. Then you've got water that goes in by and moves by diffusion, and then also osmosis. And then plants are autotrophic. What do we mean by autotrophic? They produce their own food. In other words, they're not like us. We are heterotrophic. We are consumers. We have to eat. We either eat animals that eat plants, or we eat plants that grow. So, and since we are omnivores, we eat plants and animal flesh, most of us, unless you are a vegetarian or a vegan, only eats plants. A vegetarian will eat plants and also animal matter, like they'll have yogurt, they'll eat cheese, and they'll eat eggs. And a vegan doesn't touch anything that has a face, or so they say. So it's nothing that will have a face like an animal. All right, so autotrophic, they're able to produce their own food. The word auto means on its own. Produces within the food web and provide oxygen for us, because they use carbon dioxide and give off oxygen in photosynthesis, and also they provide food for us. The economic value of plants ranges from, oh, come on, people, you should know this. You've done this in grade 7. Paper's pulped wood, fabrics, cotton, rope, which is from sisal, wicker and cane is used for furniture. Secondary growth wood is used for building and fire and fences and furniture. We know all of that. That's easy. So if you get asked, provide the economic importance of plants, there we go. Apart from providing us with food, they also provide us with a whole bunch of other things. All right, anatomy of a dicotyledonous plant. Now, dicot means it's got 2 cotyledons. Now we have angiosperms are the most highly developed plants. If we look at plants and the phyla of plants, you start off with, for example, algae, right, which is a plant because it contains chlorophyll, and that would be sort of like that and your phytoplankton, that will be at the bottom of the food chain. And as you go up through all the different kingdoms in plants, your angiosperm are your flowering plants, and they are the most advanced of all the plants. Okay, angiosperm are divided into monocotyledons. Mono means 1. Dicotyledons means 2, and you've done this before in previous years. This is just a recap, because you must know the dicotyledons. And it's always nice, you know the monocots already, so you can compare them. In monocots, your leaves are isobilateral. In other words, they are the same on both sides. Iso means the same. They have no petiole, all right? Parallel venation, and they are long and slender. Whereas with dicotyledons, specifically your angiosperm, it's dorsiventral. So dorsiventral means it's different on both sides. So if you look at the front of a leaf and you look at the back of the leaf and they are not the same, then it is a dicot plant. They have a petiole, which is a leaf stalk. They have a net venation, so your leaves will look like this. And the veins do this. And then they still branch here and they make little branches all over. In fact, it was in fashion in the 70s, and it was in fashion in the early, I think 2010, round about there, where you had leaf earrings which looked like leaves without any, that's just the veins of the leaf filled with gold or whatever, but very pretty, a fashion accessory. And then long and slender shape, and they have a variety of shapes, because your monocot, if that's the stem, the leaf wraps around the stem and then comes up, and they're long and slender, like grass, for example. Okay, whereas here you've actually got a petiole. If we look at the stems, stems are green and herbaceous, all right? In other words, they never get secondary thickening, and they only live for a year. Or maximum a year. It's normally just for a season. Lots of scattered vascular bundles, and it has no cambium present in the vascular bundle. The VB is the vascular bundle. Whereas with your woody stems, at least with your dicot stems, they are woody, and we're going to see annual rings. And then vascular bundles arrange in a circle. They're in an absolute dead straight circle all the way around the outside. Cambium between the xylem and the phloem. And it's the cambium that is meristematic. In other words, it's the cambium that's going to make growth possible. Cambium will produce xylem and phloem cells and push the xylem and the phloem, the older xylem and phloem out. All right, and then clearly, and this is an easy one to remember, you have adventitious root system for monocots. In other words, the roots do this. If this is the soil surface, the roots are very shallow and they're close to the surface of the soil. Whereas with a taproot system, it grows all the way down like that, and you'll have lateral roots that grow off that, which are mainly there for anchor and support. But that's what the taproot looks like. It's one deep root that goes deep, deep, deep down. Okay. Now, this is an example of a tree. It's a photo I found. And it is huge. Just have a look at this. This is the bottom of the tree, and these are people. So those are people sitting here having a picnic in the shade of this mammoth tree. I mean, it is absolutely huge. Now, if we look at support and transport people, that tree is going to have to get water from here, from the soil, all the way up this plant, and up and up and up and up and up and up, so that those little leaves right on the tippy end of the plant, those leaves there also get water and the nutrients so that they are able to photosynthesize right at the top here. So not only does this plant have to stay upright, okay, and not just fall over, it's got to get water right up there to those leaves. This must be about easily 20, 30 meters high. That's very, very high. Okay, if we look, we start off with the roots, and it's the roots that absorb the water. So if we look at a dicotyledonous taproot system, it's going to look like this. We've got our taproot, okay, and we're going to have lateral roots. And the lateral roots are there also. They're going to have the same as the end of the taproot, but they're mainly there to support. All right, they're to stop this thing from falling over. So those are lateral roots. Okay, now this taproot is going to have the end of it, and remember the same is going to apply to these lateral roots. It's going to have an area that slots off very easily. Now, if you were taking, guys, if you were taking a girl out on a date, and also the same with girls, in the old days, the girls wanted their hands to be all nice and soft because you're going to hold hands. So the easiest way to make sure that your hands were all soft and lovely was to take mud, and you go to the garden outside, get a handful of sand, go and make mud, and you rub your hands and you almost wash your hands with mud, okay? And what happened is it scaffs off all the dead skin and makes your hands beautifully soft. And the guys used to do it as well, so that their hands were all nice and soft for when they took the girls out. Now, you can only imagine that this poor little tip of the root here is growing in down through the sand. And sand is coarse. And it cuts and shaves away and rubs against and slots off the tip of this root. And that we call the root cap. And what grows there are cells that are quite slimy. And what they do is they help the root grow down. And as it gets slothed off, new little slimy cells form there to replace. So that as this root grows down, it doesn't damage the end of the root. Now, the next region here, so let's do lines. This region there is called the Meristematic region. And it's in this meristematic region that we are going to find mitosis taking place. So you'll have meristematic cells, and mitosis will take place to make new cells grow. That makes the root grow deeper. Okay, so you'll have new cells, and those new cells are going to push this root cap that way. All right, then we have a region here of elongation. And this is where your new cells that have been made here in the meristematic region, because remember, it's going to push downwards, those cells are then going to be able to elongate. So they go from these brand new cells with very, very large nuclei, they now start to differentiate. And so you have your region of elongation. Then we have a region here that has lots and lots of outgrowths. They're just little unicellular outgrowths. And that is the Routier region. Okay, and that Routier region is very important because this is where almost 80% of the water is absorbed at this region here. So you've got the root cap, you've got the meristematic region, you've got your region of elongation, and then you have your Routier region. This is where differentiation is taking place. And your Routier region is where 80%, just this little piece here, is where 80% of the water for the plant is absorbed. You're then going to have the same regions on this plant, and then you're going to have your Routier region. And the same here. Root cap, region of meristematic region, region of elongation, and then you have your Routier region. So the Routier regions account for 80% of the water absorption. The other 20% is absorbed everywhere else. Okay, now if we look at the internal structure of a dicotyledonous root, here you'll see that it's a single elongated section. So what we're going to have is the Routier. And it's part of one cell, so it's an extension of one cell. Then that forms part of the epidermis. And you should know by now, epi means out. So epidermis, this is an epidermis, it's the outer layer. Mesodermis is the middle layer, and endodermis would be the inner layer. All right, now you'll see here we have an endodermis. Is this red or pink that's all the way around there. We have cortex cells in the middle. Cortex cells are just a means to transport the water, okay, or a place for the water to flow through to the middle. That's number one. Number two, cortex cells are storage. Or you can also store substances in the cortex cells. For example, we eat potatoes. We eat carrots. A carrot is just a taproot. That's all it is. It's one big glorious taproot. And when you pluck it out of the ground, well, clearly the tip breaks off with the root hairs, but we get this carrot. And that is what you will see if you cut a carrot in half. So you take your carrot, cut it in half and look at it, and you'll see this structure. And the nutrients that we eat are stored in the carrot, will be stored in the cortex cells. Okay, now, in here, we're going to have the vesicular cambium, all right? And the phloem cells are always on the outside and the xylem is on the inside. So just imagine if we were naming from outside to inside, we get phloem first because the P comes before X in the alphabet and X is right in the middle. But another way of remembering it is look at the shape here. Can you see the X shape here? That X shape is for the xylem vessels. So that xylem is in the middle and the phloem is on the outside. Okay, and it makes an X shape. That's how you'll always be able to tell if it is a dicotyledonous root. Okay, now we have a line diagram here, and this line diagram is going to show the structure. So this is the one that you can expect in an exam. You have your root hair, you have your endodermis, so this outer layer of cells, endodermis, and it's there to protect. Then we have the exodermis, which is now away, it's the next layer in, the cortex cells. Then we have the endodermis, which is all the way around the vascular bundles. This is what gives the plant its support, remember. We have the phloem. I'm going to color the phloem in orange. Phloem and phloem. And then we have our xylem, and I don't have blue, but we're going to make that pink. Okay? And all of this here, these are your xylem vessels. Primary and secondary xylem. Okay? The vesicular cambium is all the way around. Let's use another color for that. Here's your vesicular cambium. And it's that cambium that produces the phloem cells, and it's what produces the xylem vessels. Okay, so you get your protoxylem is the first lot of xylem, and your mature xylem will be your metaxylem. But think of it as M for meta and the M for middle. Okay, right inside the endodermis, you'll have the pericycle, which is a layer of cells that's just inside the endodermis. And parenchyma cells are the cells that are in between. All right, they form here in between the xylem and the phloem is your parenchyma. You must know this diagram, people. Learn well. Okay, let's just recap. We have the root hair, the epidermis. Next layer is the exodermis, the cortex cells, which are also for storage and also for the water movement. Okay, then we go here. We start with the endodermis. The very next layer of cells is the pericycle. Then we have the phloem cells. We have the cambium, all right, or vesicular cambium, because it's part of vascular bundle, vesicular cambium. And then the protoxylem on the outside and the metaxylem is in the middle. All right, now, next diagram. What I've done here is I've taken a photo. This is copied from the GCSE Biology, 2nd edition, D.G. McKeon. And this is to show you where the root hairs are. Can you see the fine, this would be a lateral root, and this entire area here would be your root hairs. They are very, very small. This is highly magnified. All right, so those are your root hairs. And they account for 80% of the absorption. Now, if we look at the dicotyledonous root, the role of the cells, here we have our epidermis, and what is the epidermis going to do? First of all, it's a single layer of cells. They are thin-walled. They have a large vacuole. There is no cuticle. Now think about this, and I'm going to draw it here. Let's use pink, or no, let's use white. Okay, what we've got is that would be a neighboring cell. And then we have the Routier and another neighboring cell. Okay, here's your soils all the way around here. Okay, you're going to have, this is all soil. Then in that Routier you'll have a nucleus and a very, very large vacuole. And that vacuole is going to be filled with cell sap. Okay. Now, if you take the neighboring cell, it's going to have a nucleus, it's going to have a vacuole, etc., etc. This is your epidermal cell, or epidermis. But this here is your Routier. That's the nucleus of the Routier. This is soil. I'm not drawing this diagram like you were supposed to, but I'm just explaining as I go. And this is the large vacuole. And you're going to have a thin cell wall. Okay, and no cuticle, very important, no cuticle. And that is, your cuticle is generally a waxy layer. Now, can you imagine the root's job is to absorb water from the soil? If the root is covered with wax, is the water going to be able to get in? Definitely not. Waxy cuticles are there to prevent water loss. We're not going to lose water from the plant because the water always moves up. The minute it gets into that plant, it gets sucked up. So there's a continual sucking force. The same as if you have a glass of cool drink or milkshake and you have a straw. You suck it into your mouth, you swallow. Now immediately your mouth's got space to suck up more. The minute you let go, all of it's going to go this way. But in a plant, it's continually sucking. So water can only ever move into a plant. And we don't want to put any wax here, because if we put a waxy cuticle around it, we can't absorb the water. So there is no cuticle ever in a root. All right. Secondly, that root hair is an extension. So this extension here increases the surface area. If I take a piece of paper or a piece of string, okay? That piece of string is about 15 centimeters. If I want to fit this 15 centimeters into 1 centimeter, what do? I fold it. So I go down like that. I can fit 15 centimeters into 1 centimeter, which means I can do that 15 times. I can have something that looks like this. So in 15 centimeters, I can have 15 times 15. You follow? The same as if I have a piece of paper. Let's see. Okay, here's a scrap piece of paper. And I take this piece of paper and I let me quickly make a fan. Do you remember when you were little? Okay, boys and girls used to do this. When you were like in grade 1 and 2 or grade 0 and 1, make a fan. And I know I sometimes do it when I go to parents' evenings and the halls are hot and it gets stuffy, and what do? We make a little fan, and the little fan looks like this. Okay, I haven't folded this very nicely, but I've taken 30 centimeters and I've squished it up by folding it. And the minute there are folds, people, you increase the surface area, and I can quite comfortably fit that into 1 centimeter. So I can fit 30 centimeters into 1 centimeter by simply putting folds in it. And that's exactly what we have with the root hair. It's an extension. So instead of like a normal epidermal cell only having that surface there to absorb in, it's actually extended that surface all the way out. So you have an increased surface area. It's a single layer of cells, the epidermis. And your root hair is thin-walled, so the water comes in quickly. There is no cuticle, and it has a very large vacuole. And what this vacuole does is the vacuole is filled with cell sap. Now, cell sap is water with dissolved mineral salts. So the water will move from a higher water potential in the roots to, in the soil, to a lower water potential inside the actual root, in that vacuole. So it always goes downhill, and it will move, the water will move through the cell membrane into the vacuole, or the tonoplast of the vacuole, and into the vacuole. That will happen by osmosis. Now, what was osmosis? Do you remember? Osmosis was the movement of water, and you must know this, from a higher water potential to a lower water potential along the concentration gradient until an equilibrium, through a semi-permeable membrane. And will an equilibrium ever be reached? No, because the minute there's an equilibrium, there is no osmosis. So, osmosis is and you did this in grade 10, the movement of water from a higher water potential to a lower water potential along the concentration gradient through a selectively or partially permeable membrane. Selectively, partially, differentially all mean the same thing. All right, so if we look at what we've got here, it says modified epidermal cell region of maturation, you have root hairs, and epidermis protects underlying cells, and the root hair increases the surface area, and that is very important. The minute I take folds and there is a fold of any kind, you are increasing the surface area for absorption. So if I have my root hair, it would just stick into the soil like that. So instead of only having that much absorption, I've increased my absorption area all the way around here. So you increase the surface area for absorption so that water and mineral salts can be absorbed. Now, the next layer in the root was the cortex cells. They thin-walled, they loosely packed people, and they loosely packed parenchyma cells that function as the cortex. Now, they have, look at this, that's an intercellular airspace, and that's an intercellular airspace. So as these cells have packed, because they're rounded, you see, you're going to have spaces, and those spaces there, the intercellular air spaces that we have here, are wonderful because they help for the movement of water, okay, but also for gaseous exchange, for gases to move in and out of those cells. So they have intercellular air spaces, provides a passage for the water and the dissolved ions from the root hairs to the xylem vessels, and it stores food in the form of starch and water. Okay, I just want to take you back to this diagram. Here, the water comes in here. The water will come in here by osmosis, and it travels all the way through. Now it can go along here and all the way through and around, and it'll go through some cells and around a cell wall and through a cell until it gets to the xylem vessels. All right, so that would be your movement of your water. So what does the cortex do? It just provides a passage for the water to move through. Okay, let's just carry on here. All right, so it provides a passage for the water to move through, okay, from the root hairs to the tissue, and it stores water and mineral salts and starch. So that starch, for example, think of a carrot. And yes, guys, I know you don't do vegetables, but go and have a look, because your mother will have carrots. So go have a look at carrots in the fridge. Or go to the shopping center, and when your mom buys vegetables, just take a carrot and break it in half and have a look. That is what we are learning about here. Okay? Endodermis, and for the teachers, you can just take a carrot to school and show your learners. All right, endodermis. Now we're looking at the endodermis. It's what's inside. It's the inner layer. Now this is very, very special. people, you must pay attention here. It's a single layer of thick-walled cells. Now, thick-walled, and you think, hold on, water's got to move through there. Why are they thick-walled? Because if they've got thin walls, the water moves too easily. If they have a thick wall, why would they do this? Everything in nature has a reason, all right? Everything has a function. If it's there, it has a function, except our appendix. Everything else has a function. Okay, cell walls are covered with a sheath of waterproof suberin, okay, across a radial wall called the Casparian strip. And this Casparian strip is very important. Okay, these cells channel the water in the tissue. Now, if there's your endodermis, okay, the pericycle is the very next layer of cells around, but in your endodermis you've got this Casparian strip. And what that does is it stops the water from moving sideways. It channels the water through. So here I've got a diagram of the Casparian strips. There's your suberin, and your suberin's completely waterproof. So what does it do? It makes sure that the water coming from the cortex cells passes straight through, and it directs that water to the xylem tissue. So the water can't go up and down, it has to go straight through, into the xylem and up. Now we look at the xylem vessels. Xylem forms the vascular stele and makes the shape of a cross or star in the center of the stele. Remember, if we look at a cross-section of a taproot, the xylem will be here in the middle in the shape of an X, so you can't make a mistake. There's your xylem, your phloem is over there. Okay, the cells are of the xylem. Now everything else to now has lived. All the other cells are living cells. They've got a cytoplasm, they've got nucleus, they've got all the organelles that we have. But here, they are non-living cells. There are no cross walls. So if I look at a normal cell, I'll have a cell, and then another cell, and another cell. And these will be cross walls. Okay. In a xylem, there are no cross walls. So what's happened is we have our xylem tissue. Okay, I'm not a superb artist, but that's what the xylem tissue will look like. This would have been one cell, that would have been another cell. So what's literally happened in the xylem is those cross walls have got holes in them, and they've actually just eventually formed a tube like that. All right, and the walls have become thickened, so it makes your xylem vessels very, very strong. So if we look here, it has a very thick cell wall. This area in the middle here is called the lumen. It has a large lumen. There are no cross walls, which means it's now just become a very long tube. Okay, there are no cross walls, so it's like almost like all the insides of the cells has just been washed away. And they're rounded, which makes any round structure is strong. If something is square, you can always break the corners easily. But if it's round, it makes a round structure is always strong. All right, so let's go up here again. So they're non-living, they have no cross walls, and they form long, narrow tubes. Okay? The cell walls are thickened, and they're thickened with what? They're thickened with a substance called lignin, and you must know this. Okay? They're thickened with lignin, and each vessel has a large lumen to allow the movement of water. They have bordered pits. So if we go back to my diagram here, they're little holes. Because remember, this membrane is very, very thick. So you have these little holes in them. And those little holes are to pass water to the next xylem vessel that's over there. So the water can move into this one here, and then this one will also have bordered pits. So water can move sideways to the xylem vessel that lies next to it. Okay, so those are called bordered pits, and they allow water to move sideways into neighboring xylem vessels. All right, so xylem tissue transports, people, you must know this. This is very important. Xylem tissue transports water and mineral salts from the root up the stems to the leaves. The water is used for the process of photosynthesis, but also to keep turgidity. So xylem also gives support and structure to a plant because the cell walls are thickened with lignin. And in a leaf, the xylem is found in the veins of the leaf. All right, so xylem vessels transport water and mineral salts up the plant to the leaves so that photosynthesis can take place. Remember also that water gives the cells their turgidity. It makes the cells nice and plump. All right, and that also gives structure. So they must get water. Where do they get the water from? From the xylem. The xylem transports the water up the plant. Now the phloem. Phloem tissue is very special because what phloem does, phloem is very much like our blood supply, our veins and arteries. What it does is it transports the nutrients up and down the plant. So if we need nutrients in the roots, if we need nutrients in the stem, if we need nutrients in the leaves, it's the phloem that does it. And where the leaves are the factory during photosynthesis, they produce the food. That food has to be taken and stored somewhere. So it'll be stored in the stem, sometimes it's stored in the leaves, sometimes it's stored in the roots. So it must be taken there. How does this happen? The phloem. The phloem carries nutrients up and water up and down the plant. So this tissue consists of sieve tubes and companion cells. Now the sieve tubes are cylindrical, they're rounded. They have perforated walls called sieve plates. So think of a sieve. You'll have the cells in longitudinal section looking like this, and there'll be holes here. And then you have fibers that still run through here, but they're holes so that the liquid can move up and down. The sieve tubes have thin cell walls. They have no nucleus, because remember the liquid's moving up and down in there, and they contain strands of cytoplasm, which pass from one cell to the next. The companion cells, and these are very special little cells, lie next to these sieve tubes, so the companion cell will sit here. Now remember you're seeing it in cross section there, so you've cut through here. This, okay, so this is a cross section. And they lie next to the sieve tubes, and they do have cross walls and nuclei, and what they are living. So this here would be your companion cell. And this here would be a sieve tube. So the sieve tube will have the cytoplasm that runs through. It's got perforated edges, so that's what makes the sieve. And then next to them are living cells, and those living cells are the companion cells. And what is the phloem's function? To carry water and nutrients up and down the plant. The sieve tube transports nutrients dissolved in water, just like our blood, and your companion cells assist the sieve tubes to function. Because remember, the cytoplasm in the sieve tubes also need nutrients, and that will be supplied by your companion cells. All right, let's see how much you've remembered. Study the diagram of the external structure of a root tip and answer the questions that follow. Now, the first thing you do when you get a diagram is you label the diagram on your exam question or your test paper. Okay, so first thing we're going to do here is we're going to look at the labels. We say, right, number 1, that's the mature region. Okay, number 2, well, there are these things sticking out. That's the root hair region. Okay, number 3 is going to be the region of elongation. So, in other words, the area where it gets longer. Region 4 is the meristematic region. That's where the new cells undergo mitosis, or new cells are formed during mitosis, okay? And if we look here, number 5, and I'm going to do 5 here, 5 will be the region of growth. Why? Because your new cells are made at region 4, which is approximately there. And then you've got your little region of elongation where the cells get longer, and then they start to differentiate into root hairs and xylem, etc., etc. So 5, your region of elongation, and your meristematic region make up your region of growth. And then 6 is the root cap. And 7 is going to be a lateral root. And a lateral root's job is to absorb water a little bit, but mainly for anchorage and support. Okay, identify the regions 1 to 5, we've done that. Mature region, the Routier region, region of elongation, meristematic region, and the region of growth, because it's your elongation and meristematic together. Identify the structure number 6 and state its function. 6 is the root cap. And what does the root cap do? It covers and protects the root tip. Okay, that's all it does. It covers and protects that root tip. And that they give you 2 marks for. I mean, that's ridiculous. Okay, which numbered part consists of cells that undergo mitosis? I've told you this many times, people. It is the meristematic region. So they ask you for the number. It's number 4. And what is it called? The meristematic region. So it's number 4, the meristematic region. Name the primary function of part number 2 and name the process involved. So what is the primary function of part number 2? It's the Routier region, is to absorb water. How does it absorb that water? By osmosis and diffusion. So to absorb water. And how does it do it? It does it by osmosis. That's when the water moves into cell and vacuole. So it moves through the tonoplast, the cell membrane, and then the tonoplast of the vacuole. And then you have diffusion. Diffusion. And diffusion is when it moves in, it moves into the cell wall. So if we have our through cell walls of the cells. So if I've got my epidermal cells, and then I've got my root hair, another cell there. If the water moves in through here, it's moved in by diffusion. If it moves into the cell, then it moves in by osmosis. So remember, that's your water moving in is by osmosis and diffusion. All right? Identify the structure number 7 and state 2 functions. So if we look up here, structure number 7 is a lateral root, and I told you, first of all, the lateral root anchors the plant, and also because the root hairs are there, it's going to increase the surface area and absorb water. Then question 1.6. What part of the germinating seed gives rise to a plant root? It's the radicle. Okay, remember if I have I'm going to draw a bean seed. The little part that comes out, there's the embryo. These are your cotyledons. And the little part that grows upwards into a leaf, that's the plumule. Okay, and the part that grows down is the radicle. Okay. Now we go on to the stems. And you're going to find, people, that quite a lot of the cell structures that we have in the root are also going to be in the stem. So it's not like we're repeating it, because in an exam they can ask you either for the stem or the root. All right, not the leaf, because you did the leaf in grade 10. But you must know the leaf's importance in transpiration, or creating a suction force for transpiration. So the dicotyledonous stem, we're looking at the role of cells. The function of the stem, let's get a different color here. Let's go for pink now. What is the function of the stem? Now you know this. It provides support for the leaves, the flowers, and the fruit. Okay, it transports water and the dissolved mineral salts up to the leaves via the xylem. It transports water and dissolved nutrients up and down via the phloem. And it serves as a storage organ for reserve organic nutrients and water. So storage organ. So if we look at the functions of the stem, it's there for support of the leaves, flowers and fruit. It's there, the xylem is there to transport mineral salts up the plant and water. The phloem transports water and mineral salts and nutrients up and down the plant. And it's a storage organ. It's got to keep that. So it either keeps it in the roots or it stores it in the stems or in the leaves. But the stem is a main storage organ of nutrients. Okay, looking at the internal structure of a dicotyledonous stem, this is what we're going to have. Now this is a line diagram, it's been cut through, it's a simplified schematic diagram. So here we have, and this is a cross section, all right? I'm going to put here, it's a cross section. All right, we start off with the hair-like outgrowths now. On the root, your root hair was an epidermal outgrowth. It was unicellular. So it was one cell that had this extension to increase the surface area. But in the stem, we have multicellular structures. So if I look at this here, it will look like that. There's your epidermal cell, and the epidermal cell will have a whole bunch of other little cells that grow from it. So it's multicellular. It's got lots of cells. So that's one of the differences between a dicotyledonous root and a dicotyledonous stem. The stem has multicellular hair-like outgrowths. Then you have your epidermis. Then there's a layer of Sclerenchyma and then Parenchyma. And then there's the starch sheath. And those three form the cortex. So cortex is the name of a region. That region is made up of Sclerenchyma and Parenchyma and your starch sheath. Now you learned all these cells in grade 10. Then you have the medullary rays, which is just like ray lines, like the spoke of a wheel. You have the pith, which is the center part of the stem, and it's always the soft, mushy part. And then you have the Sclerenchyma cap, which is Sclerenchyma cells, and they are there to give support and strengthening. All right, support and strengthening. That's where I've shaded the pink. Then we have the xylem, so we'll make the xylem green. Xylem's always in the middle. Remember, when we had the root, it was in an X, so it's in the center. And the metaxylem was right in the middle. So there's the xylem, and then we'll make the phloem orange, and your phloem is there. And in between the two, we have the stuff that grows the new xylem and phloem called the cambium. Okay, so we are going to have here the medullary rays, then we have the xylem, and we have the phloem, and we have the cambium in the middle. And it's your xylem and your cambium and your phloem make up your vascular bundle, and including the Sclerenchyma cap makes up the vascular stele. Now the vascular stele, S-T-E-L-E, means the whole vascular bundle. And the whole vascular bundle will be the Sclerenchyma, making it strong, and then you have the phloem, always on the outside, the cambium, which makes the new xylem and the new phloem, and then the xylem in the middle. Always xylem in the middle. Just remember, xylem is an X. It's in the middle, always. Now, you have to know how to read micrographs. And a micrograph is a photo that's taken of the internal structure, all right, with a microscope that is incredibly strong. It's thousands of magnifications times magnified. All right, so a micrograph is you're looking at what it really looks like. Not a line diagram, but a micrograph. And what they can do in an exam is give you a micrograph and say to you, what do you see? Now, if we look at this vascular bundle here, okay, here we can see the epidermal cells. Okay, you can't really see the Sclerenchyma, the magnification isn't high enough, but you can definitely see the epidermis. You can definitely see the Parenchyma. And there's your epidermis. You can't see any trichomes or hair-like structures, which is fine. Okay, this area here in the middle, you can see how large the cells are, and they've got quite large intercellular air spaces. Right in the middle here would be the pith. Okay, here you can see very easily the Sclerenchyma. Okay, then we've got the phloem. Okay, then here the xylem. And this here would be the cambium. But it's cambium found inside the vascular bundle, so we call it vesicular cambium. It's inside, so it's vesicular cambium. But this is what a micrograph looks like. Okay, now we go from the outside in. So we have the epidermis, it's on the outside. Epidermis function always to protect. So it's brick-shaped, they thick cell walls, they have no intercellular air spaces, because you don't want anything, you just want this nice, big, strong supporting cell that is going to be there to prevent any nonsense from coming into the cell. So it's into the plant, so it's there to protect. Okay? Remember, we're doing dicot plants. It's covered with a waxy, waterproof cuticle. What? To prevent water loss by the plant. You don't want water loss through the stem. You want the stem to transport the water up to the leaves. Form the outer, tightly packed single layer of cells, and it is the outer protective covering. So you've got thick cell walls, no intercellular air spaces, a nice, thick, waxy cuticle. Now, stomata, just like with the leaves, stomata are present on young stems. So when the stems are still green, you're going to have stomata. All right. And the stomata we know are for gaseous exchange. It's so that gases, carbon dioxide can come in and oxygen can go out. Okay. Then we have in woody stems though. Now not young stems that are still green. Remember, they're still going to be green. Okay. We don't want green plants enough. Let me write that neatly. Green. Okay, woody stems, those we can't have stomata, so we have things called lenticels. And lenticels also function for gaseous exchange in woody stems, and people, you need to know that. Then woody stems, the cells below the epidermis, develop into cork cambium. Now remember, cambium is meristematic. It will always make new cells. So we get this cork cambium layer, and what happens is the cork cambium layer now produces new new cork cells. So it'll have this layer that will now produce more cork cells, so that that cork is dry and it prevents any loss of fluids to the outside. Okay, and those cork cells eventually die, and they form this protective outer layer that is waterproof. All right, so please remember, in the young stems that are green, you'll have stomata for gaseous exchange, and in the woody stems, you'll have lenticels for gaseous exchange. And just underneath that epidermis, you're going to have a layer of cork cambium, which is going to produce cork cells that die and then become corky. All right, to protect. Now, the cortex. The cortex is made up of three types of cells. We have the Sclerenchyma, we have Parenchyma, and then we have the starch sheath. Those are the three layers that make the cortex. All right, so the Sclerenchyma cells, we saw them, they're longish. They have cell walls that consist of pectin, okay, and cellulose. And they thickened at the corners, and that's for strengthening in young stems. So it's very important, the Sclerenchyma. So if we look at a Sclerenchyma cell, it will look like this. Okay, and it'll be thickened like that. Corners. They're still living, but they have thickened corners just to provide that little bit of strengthening in young stems. Then the Parenchyma, well, the parenchyma makes up most of the cortex, and they are thin cells, and they have large intercellular air spaces. Why? For storage. And then the intercellular air spaces are there for gases to exchange. All right, gases must exchange in the roots and in the stems. So those intercellular air spaces in the parenchyma are very important. And then we look at the starch sheath. This forms the inner boundary of the cortex. So it's on the inside of the cortex, and it's a single layer of cells that are tightly packed. Okay, and they're thin walls. Why do we have all these thin walls? Because we want gases to be able to pass through. Okay. And the cells have no chloroplasts, all right, but contain starch grains. Now, starch grains is glucose is stored as starch. So the glucose that's produced in the leaves will be stored there as a starch molecule, okay, and allows movement of water and nutrients to pass around the vascular bundle and the cortical cells. People, glucose will always change the water potential. You did this in grade 10. Okay, so osmosis will occur. We don't want that. We don't want the water to move sideways into the cortex cells. So what happens? It forms starch grains. And starch grains don't care about water. Water moves around it. Okay, sclerenchyma. The cell walls are, now sclerenchyma, you must know. This is very important. Why? Because sclerenchyma is support. It gives absolute total, not like parenchyma, which just is there to support your green stems. This sclerenchyma is mega. It gives, that's what, you know that tree I showed you just now? That one that was 30 meters high or 40 meters high? Sclerenchyma was part of the support for that tree. Okay, the cell walls are thickened with lignin, not suberin, with lignin, okay? And they form a waterproof barrier. The lumen is small, so it's got very little contents in the cell because the contents are dead. Very important. Sclerenchyma fibers are long and tapered at their ends, so they're long and then the ends taper like that as the next cell comes in here like that. Okay, so they're tapered. The tissue is flexible and gives the plant support by strengthening and protecting the vascular steel. So if you've got your vascular bundle, remember, your sclerenchyma sits here. It protects that vascular bundle. There's the phloem, then you have the cambium, and then you have the xylem. Okay, sclerenchyma fibers are used to make rope. Okay, they make rope, paper, and also thread. Right, because those fibers are, they're strong, they're very strong. Okay, we look at the vascular tissue. Now, vascular tissue's main job is the xylem to transport water up the plant and mineral salts, and the phloem to transport water and nutrients up and down the plant. And then the cambium, clearly, because the cambium has to make new xylem and new phloem. The vascular bundles form a ring around the inner stem and are surrounded by a layer of sclerenchyma, which is going to give strength. Each vascular bundle has xylem on the inside, people, and phloem is always on the outside on the same radius. Cambium is located between the xylem and the phloem, and it is meristematic tissue. What is meristematic tissue? Come on, think. Meristematic tissue undergoes mitosis and produces new cells. All right, and that causes secondary thickening. Secondary thickening is what we are going to see in woody stems. Now, the xylem carries the water and the dissolved nutrients up the stem only. It's only ever up. The phloem carries it up and down. So the phloem dissolved in water, think of our blood system, it's like the phloem. Cambium divides and makes secondary thickening possible. The thick cell walls of the xylem and the surrounding sclerenchyma will provide mechanical support and strength the stem. So in that stem, it's the sclerenchyma that forms part of the vascular steel and the xylem, because the xylem is so strong, that actually helps to support and strengthen the stem as it grows up. The pith was right in the middle. That's the center. Okay. The pith, also called the medulla. Medulla means middle. And is located in the center of the stem. These cells are large. They're thin-walled parenchyma cells, and they have lots and lots of intercellular air spaces. The pith stores water and starch. Intercellular air spaces assist with gaseous exchange. Your intercellular air spaces are always going to assist with gases to be exchanged, to be moved. Okay, secondary growth of woody stems. People, this is very, very important. Trees have woody stems. Okay, your dicotyledonous stems are going to grow into or have secondary thickening. For secondary thickening to take place, it has to be and have the cork cambium to stop, to have air gases moving but to not allow water through, because cork cells are waterproof. But in the center, it's also got to grow outwards. Remember, a stem doesn't just grow up, it grows sideways as well. And that sideways growth is called secondary thickening. So trees grow tall and need strength and support. So if you're looking at the xylem and the sclerenchyma. Transport tissue must also be protected. We don't want that transport tissue, the phloem and the xylem, to be damaged. Each vascular bundle is arranged in a concentric circle around the inside of the stem. So if this is the stem like that, you're going to have your vascular bundles arranged like this. Okay? The vascular bundle consists of xylem on the inside, cambium in the middle, because that's your meristematic tissue, and the phloem is always, always on the outside. The cambium located inside the vascular bundle is called Vesicular Cambium. But you're going to find cambium here. That's your Vesicular Cambium. But remember, the cambium makes a circle all the way around. So the cambium that is inside the vascular bundle is called Vesicular Cambium. The cambium between the vascular bundles is called Intervesicular Cambium, because the word inter always means between. So it's the cambium that's between the vascular bundles. Okay, now, a sclerenchyma cap forms between the phloem and the starch sheath. We know this because that's on the outside of the vascular bundle there. The sclerenchyma cap, it's like a little cap that forms there to protect the vascular bundle. That's why it's there. But it also provides strength and support to the stem. The cambium is meristematic and it's going to undergo mitosis and give you new xylem and phloem. The cambium forms the inner and outer ring of the secondary xylem and phloem. This gives wood the annual rings and tells you how old the tree is. Now, annual rings are made up of autumn wood and spring wood, whereas your heartwood and your sapwood are two completely different things. But you need to know these terms. Okay, heartwood, now think of your heart. Your heart is on the inside. So heartwood is the inner, older xylem layers. And your sapwood is the outer, younger, functional xylem. Ways to remember it, your heart is inside and it's older, so your heartwood is inside. Your sapwood carries sap, and if it's carrying sap, it's carrying water and mineral salts up. So your sapwood contains and transports cell sap. Okay, it contains and transports cell sap. The same stuff that you're going to find inside the vacuole of a cell, that's what gets transported up in that sapwood. And it's your functional xylem, which are water conducting tissue of the tree. Okay, so heartwood, the older xylem inside, and the sapwood is the xylem that actually transports the cells, or what will make up the cell sap. Okay, now, an annual ring consists of two types of wood. And the two types of wood we're looking at here, the autumn wood, okay, and the spring wood. Now, people think about it. Autumn wood is going to be when it is colder. The seasons are drier unless you live in Cape Town. So they are smaller and darker in color, and it's more compact. Why? Because there's not a lot of water. So those cells haven't had a lot of chance to grow. Whereas your springwood is going to contain xylem vessels with a very large lumen, okay? So they can carry lots of heavy flowing water up the plant from the wet soil during spring and summer. So in spring and summer, there's lots of rain, and the xylem vessels have to be able to carry that water up. So you can always see, if it's tightly packed, the cells, it's autumn wood. And if it's sort of slightly looser and you've got large lumen to carry lots of water, remember, the larger the lumen of the xylem, in other words, the hole, the more water it can carry. So that would be your springwood. Now, if you look at a transverse section, okay, transverse section, and this is of a dicot stem. There's your multicellular epidermal cell, and that's what makes up the hair. You have the cuticle. You have an epidermis, all right? Right underneath the epidermis, you're going to have your cork cambium here. It'll be about there. Okay? Then you have your Sclerenchyma and your Parenchyma, which makes up the cortex, as well as this layer here, which is going to be your starch sheath. Also makes up the cortex. All right, your medullary ray is like the spokes of a wheel from the pith outwards. That's the medullary ray. Then your endodermis layer, and then you have your open vascular bundle, because it's not enclosed, it only has the sclerenchyma tissue across the top here. So there's the sclerenchyma, okay? And you have your pericycle, which is that layer there, which is just inside the endodermis, is where your pericycle would be. And then you have your cambium. In between, you'll have your intervesicular cambium. There are your phloem cells, your xylem cells, and then you'll have your medulla or the pith. You must know this diagram and how to label it. You should also know how to draw this diagram from that micrograph I showed you earlier. Okay, now we're going to look at the transverse section of a young stem. Now, if it's a young stem, all right, you're not going to have a lot. You're going to have the sclerenchyma and a bit of xylem, but it's green, all right? So this little green stem is also going to contain chloroplasts, so you'll have collenchyma instead of parenchyma. So there's your primary xylem and your primary phloem. Tells you it is a new little stem. Okay, there's the epidermis. Okay, the very next layer inside here, now if we go back, you see you had your epidermis, and then you had your collenchyma layer starting. So we're going to now look here, and we're going to have this inner layer, there would be collenchyma, and the collenchyma will just start forming. Here, parenchyma. There's your, now, they haven't drawn the starch sheath in this diagram, but this line goes all the way through each of the vascular bundles. So we know that that is it's with a V, sorry. Vesicular Cambium. All right, that there would be the pith, or we can call it the medulla. And this little line here goes to the stuff or the line in between the xylem and phloem. That would be the Vesicular Cambium. Okay, that's your Vesicular Cambium. All right, now we move on and we say, okay, that's a young stem at the end of the first summer. So this stem is now almost a year old. So we're going to have the epidermis, we're going to have the cork cambium, making new cork cells, we're going to have the cortex made up of parenchyma, intervesicular cambium between the vascular bundles, the medullary rays, okay, like the spokes of a wheel, the pith right in the middle. Well, the pith is this area here. Okay. Vesicular cambium, this line is in, yeah, it's vesicular cambium would actually be here between it. So that would be the vesicular cambium. Because remember, this actually is where a vascular bundle would have been. All right, so this line needs to go to there. Okay, then we have the primary xylem, we have the secondary xylem, the primary phloem, and the secondary phloem. Why? Because that was made first, and then this was made second. This was made, because there's your cambium, this was made second, and this was made first. So your first layer is going to be pushed back because the second layer is now being made. Then your third layer, then your fourth layer. So everything comes from where the cambium is. The cambium, what's always closest to the cambium, will be secondary or tertiary. It's pushing that way. All right. Then we look at this, and this is at the end of the second and the third summer. So this is the second summer, that's the third summer. Look at what we've got. It's all still the same labels, people, but there is my vascular bundle. Okay? There is my cambium. Now, that would be the primary xylem, and then I'd have my secondary xylem and my tertiary xylem, if you want to get really technical. All right? And the same here. Primary phloem, and this would be my first layer, and that would be the second, and this would be the third. So the two together are going to make your first and your second year. We look at the xylem, not the phloem. The phloem gets squished up. It's the xylem that we need to focus on every single time. So your primary xylem is on the inside, and we've got two layers. So that layer is from the first year, that layer is from the second year. If we look at this one here, we've got the pith in the middle, we've got the cork cambium and the epidermis and the cortex and. But what do we have? If we look at our little vascular bundle and draw the vascular bundle, draw it in an exam or a test, draw it on your paper and say, right, there's my vascular bundle. Okay, let's see the xylem, because there's the cambium. There's 1, 2, 3. So this little stem is 3 years old. It's the third year. It's its third summer. Okay. Now this is what we would look at. You see all of these? This is how old this tree is. Now if we had to count, we don't even know if that's the center, but 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and you can carry on counting every one of these lines. When you get to here, that's all your xylem, and there's your phloem. Okay, so every time a new layer is made by the cambium, it's going to push outwards because it's going to make space for the line. Okay. Our last question. The stem of a woody 6-year-old tree, which was planted in 1992, was cut, and the width of the spring and the autumn wood in each annual ring was measured to determine the secondary growth. Study the table below and answer the questions that follow. Okay, if we look at this table, it gives you the year. So 92, 93, 94, 95, 96, 97, they're the 6 years. You've got the spring wood. So here the spring wood was only 2.5 centimeters and the autumn wood 2.5. So they got the same, very little water in summer and very little water in autumn. If we look at the width of the annual ring, we'd have to add the spring wood and the autumn wood. So that would be 5. Here, the same. So that would be 8. Here, we'd have to look at this. That would be 5. It would be 4.5. Or 4,5, 8, 5. Okay. Here, oh, this was a good year. Here it got lots and lots of rain during spring, and it got quite a lot of rain during autumn as well. So that would give me 18 millimeters. Here, this would be 8, and this here would be 5, it would be 10. Okay, so now we know what the annual ring size was. We could measure 5 centimeters, 8, 4.5. So this clearly was a very dry year, and so was that. All right, now we look at our questions. What is the difference between the autumn wood and the spring wood? Okay, now remember, autumn wood, spring wood. The autumn wood is the older wood. It's the wood that sits at the heart. Okay, so you would write autumn wood and a colon, and you would say that your autumn wood, you've got xylem vessels, form during the drier seasons or the drier season. Okay, and they are smaller, therefore they are smaller, more compact, and they, remember, they were darker in color. Okay, and then your spring wood was nice wood. That's the wood that got plenty of rain in summer and spring and summer. And your spring wood contains xylem with large lumen. Why? Because they've got to transport all this water that you're getting in spring. So a large lumen, and to carry a high water flow from rain. Okay, so in spring, lots of rain, spring and summer. Okay, so that's the difference in your autumn wood and your spring wood. Then, what is the total secondary growth of the stem? Show all your calculations. So they want to know how much this plant grew in 6 years. So I added all of those up just now, just because there were question marks there, but that's what you'd have to do. So you'd say 5 plus 8 plus 4.5 plus 1 plus 18 plus 8 and plus 10.5, and you'd add all of those up. So let's have a look. We've got 3 8s are 24, that's 34, 44, 45, 45, what am I missing? Oh, and let's start again. That's 13, 14, 15, 16, 17, 70 is 25, 35, that's 43, 53, 54. So it gives us 54. So we're going to add them all up, and they would have said show all calculations. So you would write 5,0 plus 8,0 plus 4,5 plus 18 plus 8 plus 10.5, and that would all equal to, so you go plus, 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 I'm not going to write them all out, is equals, we said 54 millimeters. Okay, and then you would have got a mark for showing your additions and a mark for 54 millimeters. If you left the millimeters off, they'd probably only give you half a mark. Okay, what is the average annual secondary growth over this period? Show your calculations. So we would take our 54 millimeters, we would divide by the 6 years, and that is going to give us 6 9s are 54, so that would give us 9,0 millimeters. Now if you have 3 marks here, they're giving you a mark for that, they're giving you a mark for 0.9, they're giving you a mark for millimeters. Okay? Do you understand why? If they say average annual secondary growth, it's over the 6 years. And it's 54 millimeters over 6 years, gives you 9 millimeters per year on average. Okay, now present the data in a suitable graph. So we're going to have to do our graph, and okay, we're going to have to add these up again. So let's see, that's 5, and that's 8, and 4.5. and 17, that's 18, and 7, that's 8, and 10,5. Now we're going to take that and we're going to put it into our graph. So remember your variables here, people. So we have this x-axis, and your x-axis is always your independent variable. It is independent of the results. You decide this. You are independent as this researcher. So you're going to say, I'm doing this over 6 years. And you decide the time period, you decide the temperature, you decide what it is you are going to do. That is independent of the results. So this is your x-axis, is your independent variable. All right? So along here, you're going to write years. And if we look at our graph, we're going to say, well, let's do, we start off with 1992, and then we do 93, 94, 95, 96. Okay, and we can go 97. So 2, 3, 4, 5, 6, 7. All right, now those are my years. That's my independent variable. Now I have to put in my dependent variable. This is going to be, sorry, I have to do this because I can't write any other way. This is the width of annual rings, okay, and it's in millimeters. Always put the amount. Now this is the dependent variable. Why? It depends on what you decide to do here as the researcher. You then have your independent variable. And here we would start at 0, okay, and we would go, have I got 20 blocks here? Yes. So we got 1, 2, 3, 4, 5, 6. No, we don't. Okay. So we'll go 2, 4, 6, 8, 10, 12, 14, 16, 18, and 20. Okay. Now these are your increases in width in millimeters. All right. Your dependent variable, independent variable. Okay, so now we have to plot. The first one was 5 and the next one is 8. So 5 would be halfway between 6 and 4. So there's my first one. So it's 5, and then in 1983 it goes to 8. And then we've got 4.5 and then 18. So 4.5 would be sort of halfway between there, so 4.5. And then it goes all the way up to 18 in 1995. Okay? And then 96, it's 8, and then 10.5. So here it goes to 8. And then in 1997, it goes to 10.5. So that would be 11, so it's sort of halfway here. All right, so if we draw our lines, it does this, and then down, and then up, and then it comes down again, and then to there. Okay, so for all of this, what is our heading? And it says present data in a suitable graph for 8 marks. What are we measuring? We're measuring the annual rings, okay, to determine the secondary growth of a tree planted in 1992 to 90, or over a 6-year period. Okay, so graph to show secondary growth of a tree planted or of a tree from 1992 to 1997. Okay, so for that, there's your 1 mark. Then the width of annual rings, 1 mark. In millimeters, 1 mark. All right, years, 1 mark. Did you put the years correctly? Yes. Did you put the numbers along here starting at 0? Yes. So before we've even started with our graph, we've got 1, 2, 3, 4, 5, 6, and 7, 8 for the actual drawing of the graph. This actual drawing of the graph only ever counts 2. In fact, most times it only counts 1 mark. What you get your marks for is your heading, okay? And you get your marks for writing your dependent variable and your independent variable correctly. Always make sure you have units. So this here would be years, and here you'd have millimeters. And then whether you started at 0 and wrote your numbers correctly so that your graph is not skewed. Okay, so this is what we would be looking at in an exam. Make sure that you can draw your graphs easily and quickly, because you get lots of marks from a graph. Reading the information represented in your graph, state in which year was secondary growth the same. So when would it have been the same? We'd have to look here, and when was it the same? Here, in 1993 and 1996. So for just reading off your graph and writing 1993 and 1996, you get 2 marks. You're looking at what you've drawn yourself. The least, provide a reason for your answer. So when did it grow the least? We know it was 4.5, which was 1994. So 1994, okay? And why? Well, we can say that the summer period was short. Okay, so there was little water, or we can say that it was a dry season. Because remember, season, because if summer was long, then it would have had a lot of time to absorb water. If the summer is short and dry, then there isn't a lot of growth that takes place. So the ring will be very short. The annual ring will be small. Okay, then the most, provide a reason for your answer. Well, that's easy. We just have to look here. It was 18, and that 18 was in 1995. So 1995. So they had a very short dry season here in 94. In 95, it poured with rain. So why? There was plenty of water available. Plenty of water in summer. Okay, it was a nice long summer, or you could have said it had plenty of water. Any one of those reasons would have been perfect. Okay, and then our very last question is, what information can be derived from the annual ring? Now people, from an annual ring, you can first of all tell the age of the tree. You can tell how old it is. Okay, secondly, you can tell the duration of the summer growing season. Why? Because remember, plants only grow in summer. Okay, they make their blossoms during spring, and then the fruit grows, and as that fruit grows, the tree's got lots of, it's got energy, it's got water, it's getting nutrients, it grows. Nothing grows in winter. Okay, in autumn the growth starts to slow down. In winter, most big trees are deciduous and lose their leaves during winter. So there they are, these bare naked things. There's no growth taking place. The trees go to sleep. Okay, so it's the duration of the summer growing season in that annual ring, because you're looking at the summer wood, okay, or the spring wood at least. And then lastly, you can also determine the quantity of water that was available. So you can determine whether it was a drought, or whether it was a very dry season, or whether it was a very rainy season, because of the size of the annual ring. The wider the annual ring, the more rain they had.